My name is Aiden Kim from Period 1 Bio, and today we will be explaining DNA replication at first. The first thing you need to know is the four nitrogen bases: adenine, thymine, cytosine, and guanine. And adenine always pairs with thymine, but cytosine always pairs with guanine, making adenine and thymine forming two hydrogen bonds, while guanine and cytosine pairs forming three hydrogen bonds. Now let's talk about the DNA helix structure. The DNA has a double helix structure, basically kind of like resembling a ladder, and the sides of the ladder consist of alternating sugar and phosphate molecules connected by covalent bonds, forming the backbone right here. Now let's talk about the anti-parallel orientation. So the two strands of DNA run in opposite directions is anti-parallel, and this is 5 prime and this is 3 prime. So since it's anti-parallel, it would be opposite sides, or it runs in opposite directions. And now let's start with DNA replication. Basically, a DNA replication is the process by which DNA makes an exact copy of itself, and it occurs during like the S phase of the cell cycle. First, when the helicase, let's say the helicase is right here and it's going through here, it kind of makes a replication fork, splitting them to two, like making them wider. And the helicase going through, and this is adenine, and this is thymine and guanine and cytosine, so they pair together. And sorry. Alright, so we could write cytosine, guanine, adenine, and thymine. And basically, the helicase enzymes unwind the double helix, and by breaking hydrogen bonds between base pairs, creating a replication fork. So like it kind of like unwinds them, and the leading strand is on the leading strands here. Sorry, this is supposed to be 5 and 3. This is supposed to be 3 and 5 because it leads here. And the bottom of this is the lagging strand. And the primase synthesizes RNA primers complementary to the DNA template, providing a starting point for the DNA polymerase. So there's like a starter, promoter, and terminator region, and it basically stops. The DNA polymerase. Let's say this is the promoter region, and this is the terminator. If the DNA polymerase knows to start here and it stops here, it basically unwinds everything here in the middle. So that's basically primase and DNA, and it creates a DNA primase. And then DNA polymerase then adds nucleotides to the growing DNA strand, extending it to the 5 prime to 3 prime direction. And now let's talk about ligase and primers. Basically, RNA primers are eventually removed and replaced with DNA by DNA polymerase, gaps between Okazaki fragments. So, and Okazaki fragments like kind of look what Okazaki fragments are. If like these are like split like this, and let's say these are like adenine and like so forth. Okazaki fragments like fill in the small chunks of space, like those, like it kind of fills it in. That's what an Okazaki fragment is. And Okazaki and then so the Okazaki fragments are sealed by ligase, creating a continuous strand. So it kind of continues, but it kind of fills the small space in. And a conservative replication is when the DNA replication is obviously semi-conservative, meaning each new DNA molecule consists of one parental strand and one newly synthesized daughter strand. So basically, it creates a new daughter strand, right here, like this. Okay, thank you for watching, and that was replication. Hello, my name is Aiden Kim, and basically I'll be reviewing transcription. So transcription is basically the process of by which the genetic information is encoded in DNA and DNA is copied into RNA. It basically occurs in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. So basically, during transcription, RNA polymerase binds to the promoter region of the DNA, and so this is the polymerase, and then I already drew it out. Sorry, and then basically it starts in the promoter region, and of the DNA, and initiates the synthesis of RNA by adding complementary RNA nucleotides like adenine, uracil, guanine, and cytosine. Basically, uracil counts now because it's an RNA, and it basically binds with a adenine, uracil, and guanine and cytosine to the DNA template strand. And the newly synthesized RNA molecule called the messenger RNA. Basically, I drew it out here, just the messenger RNA, because it unwinds while the polymerase goes all the way up to terminated region. During the middle part, it basically unwinds everything, and inside the polymerase, it basically, the messenger RNA kind of goes down and it messages other RNAs, and basically after that is then processed and transported out of the nucleus to be translated into protein in the cytoplasm. So this is the cytoplasm. Then what is it? Sorry. And then after, basically, there's a strand that comes out, and sorry, I kind of drew it out, but basically there's there's introns and exons, and the exons stay, but the introns come out. So kind of it's the opposite. So basically these little proteins, I drew kind of out, it kind of takes all the introns, and it gets separated from the exons. Sorry if you can't see that. It gets separated from the exons, and the exons go to the cytoplasm to get ready for translation. Okay, thank you and by the way, it's Aiden Kim from Period 1. Thank you. Bye for bio. Hi, my name is Aiden Kim, and today from Period 1 Bio Honors, and today we'll be explaining translation. So translation is basically the process by which genetic information is encoded in messenger RNA, also known as mRNA, and is decoded to produce a specific sequence of amino acids. So this is the amino acid, and this is the codon, and this is the anticodon, and this is the codon. Basically it's different because it picks up the RNA, and it kind of transfers, and another anticodon comes that matches the cytosine, guanine. So basically guanine, and cytosine. So it kind of comes and matches, and this process occurs at the cellular structures called ribosomes, which are composed of ribosomal RNA and proteins. So basically translation begins with the binding of mRNA to a ribosome. And the small subunit of ribosome attaches to the mRNA molecule. So basically, this is like the it initiates, and then basically this is, I told you again, this is the anticodon. It picks up the same codon, and it basically codes for amino acid and for other things. So this goes, and then a new one comes in. Here, I'll draw it real quickly, and then let's say this is carrying GGC, because it should be carrying, so it matches with these, and this is the A site. So this goes to the A site, and this goes, and the ball comes here, and it keeps stacking up until it comes to the terminator region. And during elongation, the ribosome moves along the mRNA, which is the messenger RNA molecule, reading each codon, and matching it with the appropriate tRNA molecule carrying the complementary anti-codon sequence. Basically, just what I explained, and just like rephrasing it, and then translation continues until a stop codon, which is like it could be like UAA. Let's write it here. UAA, which is like uracil and adenine. That's like where the codons know where to stop, and is encountered on the mRNA molecule. When a stop codon enters the ribosome's A site, it signals the end of translation. So it stops. And the balls leave, and release factors bind to the ribosome, causing the newly synthesized polypeptide chain to be released, and the ribosome subunits to dissociate from mRNA. So basically, during translation, the ribosome acts as how should I say this? Molecular machine, facilitating the interaction between mRNA and tRNA molecules. So basically, that's like translation, carrying out other information, and that will be translation. Thank you for watching.